So it's this. First of all, thank you all for coming and for braving the weather forecast, which predicted rain. So obviously all of us expected rain. So here is the image which I like and which summarizes what I told you previous time about different scales of organization of DNA in the cell. And today, specifically, I don't want to talk about this small scale or this scale or this scale. I want to concentrate on this question. The largest scale, the most coarse-grained picture of all, which is chromosome territories. So I told you before that this is a big surprise because normal polymers don't want to, they don't like it. They are not supposed to behave like this. They are supposed to instead to mix together. So why would it behave like this? I told you also that and this is some surprise for me. I recently learned that, in fact, this territorial structure is even observed in some viruses, such as influenza virus, where genome consists of several RNA molecules, and they apparently also segregate. I don't have any theory for this. It's just an interesting observation which invites thinking and waters my appetite. What could possibly be the reason for such a territorial segregation of these RNAs? I don't know the answer to this. But I told you that for nuclear DNA, for DNA in the cell nucleus, the most informative of all is the high-C method. And the least piece of information that you can extract from all these hugely informative high-C maps, which tells you probabilities of meetings of different things, the least information that you can get is the probability that two loci, which are genetic distance s apart, that they meet in space. This is to extract from this hugely informative map, extract this piece of information. I like to compare it with hammering a nail using your laptop. But this is the least you can do. But this least is nevertheless interesting and important. And you see that from that point of view, these higher organisms behave very differently from lower, or better to say, organisms with longer genomes behave very differently from the organisms with shorter genomes. My understanding of this difference is that of reptation time. And I already at this stage, I want to hint to you that this difference, I think, is related to the fact that reptation time goes like the length of the polymer to the power 3.5. And if the length of the polymer is different by 2 orders of magnitude, it leads to 7 orders of magnitude in reptation time difference. And these guys do not reptate, and these guys do reptate, and that's the difference. But that's a hypothesis. For now, just let's concentrate on the fact that this fall off, this probability of meeting of two loci decays slower. If you have questions about the stuff that I'm talking about, you better ask me. I am prepared to try to answer. Okay. So the question is, how can we construct a polymer model that will exhibit territorial behavior, that will exhibit some territories, and slow decaying probability of meeting to units, to monomers? How can we construct the polymer model that satisfies these two conditions simultaneously? So there are several candidates for this task. And the first candidate that I will review is the idea of a conformation of a polymer when it collapses, but collapses rapidly, such that it does not have time to relax. It collapses very rapidly. And by the way, I told you previous time that experimentally, if you force polymer chain to collapse by dissolving one polymer chain in a liquid droplet and then allowing the solvent from this droplet to evaporate, then the droplet shrinks, and collapses this polymer chain quite rapidly. So what happens if polymer chain collapses rapidly? The analysis of this question started, as many other things, started from Dejean. Dejean thought about dynamics of chain collapse. This is his drawing, I guess. At the time, maybe it was drawn by hand, I'm not sure, but that's from his paper. And his idea was that it's a hierarchical process. If you take your chain and place it in the solvent where monomers tend to glue together when they meet, then close neighbors on the chain are more likely to meet. They form sausage like this, and sausage forms super sausage, and so on, until this whole thing becomes spherical. Now, if you think about it, this self-similar process cannot lead to equilibrium collapsed chain, because equilibrium collapsed chain, just like many different chains, just like my spaghetti in this picture, pieces of the same polymer should behave pretty much like these different spaghettis. And therefore, each piece has to go as Gaussian chain from one end to another, from one end to another. And this is not what you obtain here. So it does not lead, this process does not lead to equilibrium globule. It leads to something else. So in this drawing, we try to follow the idea that it's self-similar. This piece collapses in itself, then several such pieces collapse together, and so on. And you obtain some self-similar object, which we can call crumpled globule, because it's a result of rapid crumpling, and it's a self-similar object. It was invented by these two people when they were both students. It was a long time ago. In reality, it turns out that the process of this collapse is more complicated. So here is the most sophisticated computer simulation of this rapid collapse situation. And you see that instead of this sausage, you have something like a pearl necklace, or you have some pieces which form clumps, and when the material accumulates on these clumps, it makes the chain between clumps stretch. And this stretch chain, so it becomes a complicated process, really. So how does it work is not completely understood, to the best of my knowledge. The most recent paper on this is 2015, almost 10 years ago, but still there was no complete understanding of this subject. So the idea is probably right that there is some, I would say the idea that this self-similar process leads to self-similar structure, that idea is right. Probably rapid collapse of polymer chain does lead to something similar, but what specifically, it is an open question, not completely resolved at this point. Soon after this self-similar scenario was invented, Itzhak Rabin and a number of others suggested that it's probably relevant for DNA in the nucleus, but that was just a hypothesis. Okay, so that I told you already, that indeed, if you have such a rapidly collapsed polymer chains, then they do tend to separate from one another. Not only they are self-similar inside, which means different parts like this remain separated from one another. But chain from chain also remain separated. For some time, it doesn't mean they will remain separated forever, but for some time, and maybe a long time, they remain separated. Okay, so this picture I showed to you, yes? There is a question, yes?
My question is, so you've looked at the collapse of a single chain, and you conclude that multiple chain would collapse in a separate way. I'm not getting the next step.
Okay, so my argument went the following way. If you consider a collapse of a single chain, it leads to self-similar object. Now, if you take these rapidly collapsed chains and bring them together, then, as this simulation picture shows, they remain segregated for some time, which sort of suggests to me that this self-similarity that exists inside such territory, inside one chain, this self-similarity sort of wants to propagate further. But of course, there is no self-similarity really at larger scale because all of these chains are the same. So all these territories are of the same size. That's the logic. But basically, the idea is that the same physical mechanism that prevents this little piece to mix with surrounding little pieces, the same mechanism prevents this violet blob to mix with the green one. Okay? So this picture basically shows self-similarity in two different ways. And the only reason I show this picture to you is to illustrate to you that the science does make a progress. Look at the quality of illustration of 1988 and 2009. In 20-something years, the quality of drawing underwent dramatic improvement. But the idea is still identical. So if you think about the mathematics of this, mathematicians thought about this type of objects, calling them space-filling curves for the last over a century. And it started from Peano and Hilbert. They started, of course, in 2 dimensions, constructing space-filling curves in 2 dimensions. Of course, in mathematical literature, the main question is about how to construct the limit. How to construct the limit when the number of for instance, in this 3-dimensional case, I show several steps of this fractal iterative construction. And in mathematics, they think what happens when the number of these steps goes to infinity, and the rest, it has to do with foundations of calculus, the nature of numbers, and all of this. I do not go there because, of course, my polymer chain always has some thickness, and I don't have to do too many of these steps. But you see that there is obvious self-similar construction of this 3-dimensional Hilbert curve. So you may ask if this Hilbert curve is similar to what happens if polymer chain undergoes a rapid collapse. Notice that when I color this polymer chain in rainbow colors from one end to the other, then I obtain indeed territorial segregation of this object. It's territorial. This Hilbert curve is territorial in this sense. Okay? So this also illustrates, this is a picture I took myself in some forest, and obviously there was some insect that was reading papers by Hilbert and Piano and trying to construct a self-similar space-filling curve quite successfully, I would say. Okay, so this is one candidate. Basically, all these ideas are about one candidate for the polymer model that satisfies our two criteria. It is territorial, and it has slow decaying POS. One candidate. Now here is another candidate, another potential candidate. And previous time I spoke for too long, and therefore I will carefully watch the time in order to avoid this mistake. So another candidate, another potential candidate is obtained if you remember that the problem with the previous candidate was chain ends. It was never clear what happens to chain ends. Chain ends is a shaky issue, how ends are behaving. So let's get rid of ends. Let's consider rings. Okay, topological rings. And this turns out to be an interesting study in its own right. Whether this is a faithful model to represent chromosomes or not is another question. It's a different question, but it's a very useful and interesting model in its own right. So let's look at them. So the simplest thing that you can think about rings is to consider one ring in the presence of an infinite spike that goes perpendicular to the page through this place, and then obviously there is topological constraint. The spike is either pierces through the ring or does not. Or maybe it pierces a couple of times or a few times. So for instance, you can consider what is the virial coefficient of interaction between ring and the spike and so on. So this model is a beautiful model, and I recommend reading these papers and later papers because it's a beautiful mathematical model related to electromagnetism and everything else. Next step, you can consider two such rings, and again, there is topological constraint because they are either unconcatenated like this or then maybe concatenated like this. When you consider unconcatenated rings, that means you exclude all configurations of this type, and that means, for instance, if you have a gas of such rings, they interact even though they may not have any other forces except forces prohibiting their crossing. No other forces, nevertheless, they interact for purely topological reasons, and you can observe this if you measure, let's say, pressure of the gas of such rings. And it is indeed observed not only in computer experiment, but even in the real experiment. You can also consider the interaction of parts of the same loop, and this one was initiated by Jacques de Cloiseau. But I want to concentrate on something else, namely on the concentrated system, when you have many loops squished together. So this is similar in a way to this model, except this is made of linear chains, and I want to squish together rings. And I will claim that rings will remain territorial forever. If they are unconcatenated, they will remain territorial forever. So rings, by the way, are important for another reason that I mentioned previous time, that this extra chromosomal DNA that exists in many cancerous cells is represented by the rings, unconcatenated rings, and they are in post-solvent, so this is very relevant from that point of view too. So one thing that you may consider is you may consider squishing one ring in a closed volume. And indeed, you discover that if you do not insist on the idea that these pieces of the ring do not cross each other, and you allow your ring to cross, which means you allow your ring to be any knot it wants, then you obtain some uncharacteristic, uninteresting mixture. But if you force your ring to be an unknot like this and collapse it, squish it, it is territorial. So it's a good sign that we are on the right track. So here is a simulation performed by these two gentlemen, and what it shows is the following. In the simulation, on the left, you simulate melt of linear chains. These linear chains behave like my spaghetti. They obey Flory's theorem, and they interpenetrate. You see only one linear polymer chain, but in reality, all this black background is full of other chains that move together with this one. And you see that this is indeed a linear chain, pretty much like a Gaussian chain. Here you have similar movie for a melt or concentrated system of different rings, unconcatenated rings. And you see that each ring, and you see only one ring, but there are many other rings around, and you see that each ring is collapsed. So it's territorial. You see immediately by your eye that this is territorial. So if you do it more carefully, in more sophisticated way, you discover that indeed, the beauty of this picture, there are many models, many simulations, the lattice of lattice, all of this. I do not go through this, and this all collapses together. And when the length of these loops become very long, then you obtain this power law, which exactly corresponds to each ring being collapsed, its size goes like n to the 1/3. It is a bit complicated to read this graph because for some reason this is all normalized in a peculiar, tricky way. But I will not go through this technical detail. The fact of the matter is that there is very broad crossover, but in the end of the day, it all collapses to the power and 1/3, which means territorial behavior here. Now, the beauty of it is that in order to collapse all this data, you have to renormalize, you have to rescale the length of the polymer by the so-called entanglement length. Entanglement length is something that was introduced by the fathers of modern polymer science, Dejean and Edwards, in various combinations in a complicated history. And it is, in fact, sort of a stroke of genius, this concept of entanglement length. So I here suggest to you a simple illustration why this idea of entanglement length is so sensitive. When people in lectures for students, people explain the nature of entanglements, people say, oh, invariably, every lecturer that I heard say, oh, look at this. This is, of course, entanglement, right? These two pieces are entangled, like my picture on the left, they are entangled. These two are not, okay? So, all right. So, yeah, maybe, but if you want to move them in opposite direction, then becomes shaky, but even more important, how about this? This is not entangled, is it? This is not entangled. How about now? Is it entangled? Yeah, blue and red are now entangled. So that means entanglement, although this picture is so appealing, entanglement is not a two-body effect. Entanglement between two depends on who lives around here. So the idea that there is one scale of polymer length that characterizes the distance between something that is like this, and that's a true stroke of genius. And this stroke of genius helped when we measure this entanglement length for every model that we are dealing with, and rescale all polymers lengths by this entanglement length, then we obtain this collapse. That means, indeed, in this territorial structure, entanglements are playing controlled role. They control the whole segregation between territories. And that means entanglement length is the king of the game. In particular, we will have to discuss what is, if we really believe all of this, what is the entanglement length for chromatin fiber? What is it? How to measure it? What is the numerical value of it? And we will come back to this. Okay, so one thing that you may try to do if you want to characterize these territories, this structure of these territorial polymers, which are rings in this case. One thing that you may want to do is, since they are rings, you can construct a soap film for each ring, a minimal surface that you place on each soap film. You can do it computationally. It is tough, but possible. You obtain surfaces which are pretty much like this. So the border of this surface is this ring polymer, and those are territorial polymers. Each of them is territorial. So notice that it is quite a complicated surface, but if you measure the surface area of it and plot the surface area as a function of polymer chain length, you divide the surface area by n, you pretty much discover soon, and there are probably better data than this, but it's just the first publication on this subject. So what you discover is that these surfaces are proportional to n. That means your normally you would, n is kind of circumference, so it more or less the surface, if you have circumference like this, the surface would go like circumference squared with coefficient related to pi and stuff, if you have a circle like this. So the fact that area is proportional to n indicates that it's more or less folded in two, with very narrow gap between the two parts. Of course, it's not linear. It is folded like a branched object. Sorry, my slides were ordered. When I speak, my thought develops in a certain way, and when I was preparing, my thought obviously developed in a slightly different way. So that gives some disconnect in the order of slides. So I was saying that polymer chain shown here in green is folded in two, like this. It's a branched object folded in halves, and this corresponds to the Edwards idea of polymer in the lattice of obstacles, and so on and so forth. Michael Rubinstein and his co-worker invented a better model in which they emphasize that it's not quite like quite folded in two like this, and there are some openings, and indeed computer simulations show that there are penetrations. Some of these branches penetrate into neighboring stuff and so on. So there are lots of details about it. And people, if we have time, I will show to you some experimental data on how people study these rings, and many things are known about this. But some things remain unknown, and here I want to emphasize something that is unknown. What is unknown is the following. I told you, all of this tells you that territories, okay, with squished rings we obtain territorial polymers. Okay? And similarly for rapid polymer chain collapse, in some form we probably also obtain territories. So territories, we sort of have territories. But now we also want slow decay of this contact probability. Remember, we have two components, territories and slow decay of probability. So we have territories. Do we have slow decay of probabilities? Well, yes, we do. But how slow is slow? The slowness of this decay, you can think of this as this contact probability of subchain goes like a chain length to the power minus gamma. So you want this gamma. You want to know this gamma. So pay attention to theorem number 2. So Jan Smrek, when he was a student, proved the following theorem. And when we started working on that, Eretz, Lieberman, Eiden provided very important push and hint and so on. So his contribution was super important in that. The idea of the proof actually came from the drawing of a Chaire, when you have space-filling mosaic of some horses or some, I forgot what else, birds. You know, there are beautiful drawings by Chaire. So Jan was inspired by that and constructed and proved the following theorem. He said explicitly, by construction, he constructed territorial space-filling curves with any value of gamma, any power, in the limit between 3/2 and 1, you can construct for any desirable power, you can construct these space-filling curves. Okay? So 3/2 is trivial. It's Gaussian chains that go from one end to another and come back and so on. This is trivial. 1 is much less trivial, but okay, and you can construct anything in between. And as I said, the idea of the proof is to construct this Chaire type drawings, space-filling drawings, here shown in 2 dimensions, but he was able, I am unable to think in 3 dimensions like this, but he was able to construct similar Chaire type drawings in 3 dimensions, and proved by construction that you can have space-filling curves with any power. And now the question is, of course, that of entropy. Remember, I told you previous time about entropy of high-C maps, and here is again the same question of entropy. What is the entropically dominant? In other words, what is the most typical, statistically most typical, most probable type of conformations, and what kind of exponent, and by extension all other exponents, will be for most probable conformations of these fields. The answer to this question is I don't know. I don't know the answer to this question, and as far as I know, nobody does, but there are some speculations on this, or some preliminary arguments about this, and they are the fruits of our recent discussion with Kirill Polovnikov. So we discussed this question in connection with the Clouzot ideas that I was speaking about a week ago. So how am I doing? I am now 45 minutes past nominal beginning time, but I...
You almost have 30 minutes.
Okay, 30 minutes, I see, okay. And then I have time to drink some water. Okay, so let me walk you through this argument. So this P of S probability that two units separated by genomic distance S meet together in space. This is P of S. So it is a power law like this. We assume that this is a power law like this, but obviously it's only a power law for rings. It's a power law when the distance between them, genetic distance, is smaller than the whole length of the ring, because otherwise you have to worry about the second half too, right? But if you consider not situation of relatively close to loci, but arbitrary to loci, then the scaling form will have to be like this, with some function of S divided by N. And then for two on the ring, if you consider what is the probability that two for instance, monomer 0 and monomer 1/2 n. What is the probability that they meet in space? That probability must go as the power of n of total length of the ring, with the same power gamma. I call it gamma contact for some historical reason, but it's same power. Now let's imagine the Clouzot estimate. The Clouzot told us, I explained this to you previous time, the Clouzot told us that basically, if you go through the Clouzot argument, then this is your estimate for how it should go, where theta is the Clouzot index. And now what Kirill guessed, and supported by delicate arguments, that this theta, the Clouzot index, for rings should be the same as for linear chains. So this is basically the idea that two pieces of the ring, when they come close together, these pieces, the correlation structure in the correlation hole around these two pieces does not care about the fact that they are at a ring and so on. Interestingly, this probably works only under the assumption that the whole ring is not a knot. Because if the whole ring is not a knot, then when two of the units come together, you obtain something like this. So your high C experiment, your formaldehyde, glues together these 2 points. So this is not a knot, this is not a knot, and this is not a knot. Right? So it's believable that this process will be self-similar. While if this thing was a knot from the very beginning, then this is one knot and this is a different knot. and then you lose self-similarity and it doesn't work. So these technicalities apart, if you believe, Kirill, that indeed this theta should be the same for these rings as for linear chains, then you discover that this power gamma, you use their clause, beautiful epsilon expansion results, and so on for the theta, and you obtain 1.17, while simulations, the best available simulation, give 1.15, which is unbelievable accurate agreement. So that seems to be, we seems to be in business. And of course, this deserves much more testing in simulations. Again, this may or may not be related to chromosomes, but it's an interesting physical question in its own right.
So you use the de Cloizeau argument for a chain in a good solvent? Or in
Yes, it's the Cloizeau value of this theta for chain in the good solvent. Assuming that inside the territorial structure, because it's territorial, there is no screening, and the correlations which de Cloizeau was talking about are still the same. That's the argument, that's the idea. Whether it's valid or not, it's very much subject to further thinking, further testing, and so on, but I am very much impressed by the beauty of argument, so I decided to share it with you. Okay, so there is this story about rings, and it looks like, by all indications, squished rings behave very differently from squished linear polymer chains, which must be tested in experiment. So here is the movie which suggests the experiment. It should be, the movie should be here. No, it doesn't work. It may actually be that the movie will not run. Okay, so now I think it works. Okay, so we first take spaghetti, and then you take this ramen. You first cook spaghetti, and then you try to pull out spaghetti out of the, and it doesn't want to get out. You know, that's why I never order spaghetti in any restaurant, because I don't know how to eat them. I pull it out, and the whole thing pulls out. Okay, so if you instead cook this thing, ramen, you obtain something, it pulls out much easier. Okay, so that's the difference between, I don't think I will have any more movies, but I thought that this movie is nice, created by Jonathan Halverson. Okay, so I must also mention that there is another mathematical model which sort of reproduces all this territorial behavior and the slow scaling of contact probability and all these details. It reproduces all of this in a very beautiful way, but also, to my taste, in a very artificial way. So the way the model is constructed in this particular paper by these authors, the model is constructed from harmonic springs. Everybody interacts to everybody by a harmonic string. So, for instance, this monomer interacts along the chain with two neighbors along the chain with harmonic springs. That's the usual bead spring model of the polymer. That's the usual stuff. But now it interacts with second neighbor, third neighbor, fourth neighbor, and so on. Interacts with through springs of different spring constants, right? And you assume that the spring constant for the second neighbor, third neighbor, fourth neighbor, and so on, they form hierarchy. They depend on this genomic distance in some properly chosen, guess what? Properly chosen power law way. And if you cleverly produce this power law, then you generate polymers with desirable fractality, with desirable POS, with desirable territories and everything. Since it's Gaussian, you can compute everything and so on. I call it spooky action at the distance because I don't know what is the physics behind this particular spring. It's sort of self-similar, self-consistent description of reality. But the advantage, I mentioned this because here you can compute everything. So this is a very useful model from that point of view. So, okay. So to summarize this story, we have a few candidates for polymer model that is capable of reproducing territoriality and slow decay of this POS. We have several such models. And it seems that these models, these systems somehow speak to each other. And it seems that different properties, territoriality, slow decay of POS, they seem to go together in this model. Also, fractal dimension 3 of the chain backbone, or fractal dimension greater than 2, for sure, of the chain backbone. And the fact that reputations are somehow either excluded because time is too short, or they're excluded in the rings because there are no ends and repetition is impossible. And there are no knots at all, or almost no knots. All these things seem to go together. So the intuition would be that if you somehow prohibit repetitions, or if you somehow insist that there are no knots, is that true that if you have no knots and squish together the systems that have no knots, then you obtain territoriality, fractal dimension 3, this and so on? Is that true or not entirely true? I do not know. It's an interesting open question. These examples that we studied seem to suggest that all of these issues are closely related to one another, and probably each one of them is sufficient to generate all the others. But I'm not sure. But we have considered quite a few of these systems. We considered a mathematical space filling curves. We considered a rapidly collapsed polymer chain, rings in the melt, one ring in the cavity. I did not mention this to you, but in the cavity you can impose regular boundary conditions, just non-penetrable bound conditions. Or you can say that there is periodic boundary condition. So the loop that goes outside of this side enters from this side. So it's periodic boundary. It turns out that the result is significantly different. So these territorial structures, they are apparently very sensitive to boundary conditions, which is not entirely understood. And there are quite a few other spooky actions. At the distance, these Schmreck curves, which are similar to Escher drawings and so on. And there are many names of all these things. You see, crumpled globule, fractal globule is something that people who invented high C and observed something like this in real nucleus, they invented the word fractal globule. Loopy globule is something that Michael Rubinstein invented and preferred to call it loopy globule. Lattice animals. By the way, my favorite fact characterizing the level at which physicists understand biology is the fact that lattice animals are frequently called trees. So there are annealed lattice trees, annealed lattice animals. Also, there are marginally compact. Italian colleagues, of course, invented ring spaghetti, which are called annelloni. The word annello, as I am told, means ring in Italian. So my attitude to this is that probably my hypothesis is all of these are different names of the same thing. All of these are different realizations of the same nature. I hope that there is one universality class, and all of these guys belong to the same, they are basically the same. It's a hope, it's a hypothesis, there is no proof at this point in time, but overall, my feeling is that we as a community are much more successful inventing various names than understanding their meaning. Okay, so to summarize, high CE and territories, high CE and territories speak together, and if you construct polymer model that has no repetition for one reason or another, it's suppressed repetition because of slow time, or it's suppressed because there are no ends, or you can do a variety of things, you obtain territories. If you allow repetitions, you don't obtain territories. And so this is repetitions, there is no repetitions, this goes hand in hand. This is my major message to you. Now, this is just a beautiful image. I think, I don't know who drew it. Maybe some artist like Mondrian or somebody else could have drawn it, but it's just a piece of modern art. Now, the rest, I have a few slides about experiments on rings. So I can tell you about this, or I can stop at this point, and if you have questions, I'm happy to answer questions. Yes, please.
Really naive question, I'm sorry. So in what you talked about today, I didn't see much dynamics. It was mostly statistic properties, I think. But you insist on rotation or no rotation, which is a dynamic phenomenon, if I understand correctly. So can you explain why and how do you prevent rotation, or how one would prevent rotation, and why is rotation compared, should it be compared to some other dynamical process that you haven't talked about to know whether there is or there isn't?
This is a very good question, thank you. In fact, I did not forget about dynamics, because my plan was to speak about dynamics in next week and the week after. Those were the plans to speak about dynamics, which is why I did not speak about dynamics here. But yes, in brief, if you allow chains to replicate, then, and they have ends, then obviously with time, when the repetition completes, they will destroy all this territoriality and everything else. There is no doubt about it, because the repetition will help establish equilibrium state, and the equilibrium state, we know it's Flory theorem and all this. So that's for sure true. It is also supported directly by computer simulation. If you consider these squished rings in the melt and cut them computationally and allow to replicate, then all these territories dissolve. Okay, so there is no doubt that repetition will destroy this. How rapidly is a different question, but yes, repetition. Are there other dynamical processes that can destroy this the answer is no, because we showed by computer simulation that it stayed there forever. And we have some theoretical arguments to support this idea that without repetition, any other dynamical process that you may have, short of action of topological isomerase, that is a separate issue. So that's my attempted answer. Any other question? So Jean-François, look at the watch and tell me, should I go through this?
5 to 10 minutes more.
Okay, all right, so let me tell you. There are some interesting things about these experiments with these ring polymers. So this part, although illuminating, but does not have any direct projection. That's maybe doesn't necessarily have direct projection on the chromosomes and chromatin. So how do rings behave in the flow? If you just flow solvent and flow the liquid of polymer liquid. There are two different types of flows that I want to consider. Shear flow and this so-called extensional flow, when you pump your fluid through the narrow part of the pipe. Okay, so what happens in the extensional flow is quite interesting, because it turns out that in the extensional flow, so this picture is the result of computer simulation by this group of people, and this picture is the result of my real experiment. I bought in supermarkets some vegetables or some boxes that were wrapped by rubber rings, and I connected these rubber rings in pairs like this or in triplets like this. So they observed in simulation exactly the same process. When this set of rings is subject to extensional flow, they can connect like this, and they form super chains of such connections. These super chains are macroscopic in length, and the whole liquid becomes like reinforced concrete. When you try to pull it, instead of just flowing separate rings past one another, there is the rope that goes from one end to another and prevents any deformation. You cannot do anything. So this is what happens to the set of rings. Presumably something similar may happen also if there are not rings, but on some level to linear chains before they reptate out of such entanglement. When you subject your rings to shear flow, and here I show also geographic map, because the experiment on the shear flow of ring polymers, the most important center for this experiment is an unexpected place for most of the people. It's a place for vacation, not for work. And there are people who work there. Dimitris Vlasopoulos is a professor who works in the university there and performs these beautiful experiments. In particular, what happens if you subject rings to the shear flow. And roughly what happens to them is they basically fold in two. And each of them behaves like it is confined in a slit. In the Jean's textbook, there is scaling exercise where he considered what happens to polymer chain if you squeeze it between two plates. So this ring, for instance, this red ring, behaves as if it is confined by the forces of the shear. And this is picture from simulation, of course. So let me skip the details on how the theory of it works. And let me just show you to you the summary of the result. So basically, what happens is you have this is shear rate. Gamma dot is the shear rate, and it's normalized properly. And this is viscosity, also normalized by the shear rate. So when you have extensional flow, I showed to you in extensional flow, there is this reinforced concrete effect. So viscosity shoots up. It becomes very large, almost infinite. Here, viscosity goes down. It's shear thinning with shear rate, viscosity goes down because these guys are squished and viscosity goes down. And so the main point of this picture is to show to you that these symbols, which are the results of simulations, these symbols, which are a result of real experiment, and the theoretical curve, all of them agree. So indeed, this indicates there is a formula which I will not comment on, but this all means that indeed there is this shear slit picture, and you indeed observe rings which are squished like this. So, as I said, this is not necessarily related to chromatin context, but if territoriality is there, then probably we should understand the rings themselves properly. So that's why I told you this story. Okay, I stop. Thank you.